This is a lecture on skeletal muscle tissue. I believe we all remember this gentleman. He was our governor. And look at him. He has lots of muscles on his body. We can see the big pectoralis major muscle here on the left. We can see the pectoralis major on the muscle right. When I say major, there will be minor, pectoralis minor, which we cannot see because pectoralis minor is just hiding underneath the big pectoralis major. And we can see the big biceps muscles, and we can see the big tricep muscles here. Look at that. Such a wonderful physique. And if we look in our thigh, like in the front of the thigh, we have a big bulk of muscles, which we call quadricep muscles. Quadricep muscle, it is say like quadri mean four. So there are four muscles involved, which are vastus medialis, which is just here, vastus lateralis, which will be somewhere around here, rectus femoris here, and vastus intermedius, which is just hiding underneath the vastus lateralis. So now we can see the muscles for the thigh, which is in front of the thigh. And now we can see like his latissimus dorsi muscle here. So who start talking about these muscles? Who start studying about these muscles to the microscopic, very microscopic level? So these are the scientists which we need to know. The first two scientists, Draper and Hodges, they are Australian scientists. They start looking under the very detailed microscope and start talking about the sarcomere. And now the other scientists, Gene Hansen and Hugh Huxley, they will be talking about the gliding of the myofilaments in the muscle. So I can only find this photograph. I cannot find three other photographs of these scientists. These are the papers they have written about the study of the muscle with the electron microscope. So Draper and Hodges, they are the Australian scientists. So look at that again, like we can see the big bulk of my biceps, big bulk of triceps, and then trapezius, pectoralis major muscle. So now we have three types of muscles. The muscles we were looking at is the striated muscle. Here we can see lots of striations, and they go and attach to the joint and they move the joint and bones, and therefore they are called skeletal muscle. This is a cardiac muscle. As you know, they have some striations, but they do branch and they do join. And these are the smooth muscles, which are like a spindle shape, multiple layers. And sometimes, if we are talking about histology slide, I might confuse with this a connective tissue, which are the nucleus, like little cells floating in the sea of collagen fibers. So now let's go and study the skeletal muscle cells, which is striated muscle. So this is the muscle under the electron microscope, which we see. Now we can see two little Z line, black line here, and another Z line here. And there is a 1 center little black line, which we call M line. So the basic unit, that's what we are seeing, the basic unit of the muscle, which is what we call sarcomere, which is in between 2Z. And the structures in between 2Z And this structure is called sarcomere. So if you look at that electron microscopic picture, and now we are looking at this diagram, I'm going to show you again. 2 Z line with the M line in the center, or the fibers running from Z one side to other side. If we look at that, now we can see the orange 2Z line and with fibers running from one Z to another. Now we start seeing two different types of lines, the big, thick myosin and thin, small, little actin lines. If you look at that, myosin is in the middle going across the M, but they do not directly attach to the Z line. If you look at the actin, actins originate from the Z line, but they do not attach to the M line. And they go alternate, actin, myosin, actin, myosin, actin, myosin, actin, and so on. So this is the simpler drawing diagram of this photograph. So we were talking about cell, talking about tissue, and we know that a cell is the structural unit of living, and a tissue is the group of cells with same function. And we know that there are 4 types of cell in our body, which are epithelial, connective, mast cell, and nervous tissue. And today we are focusing on the mast cell tissue. So when we say a tissue is made of many cells, cell plus cell is equal to tissue, which means squamous cell plus many squamous cells, we make the squamous epithelial tissue. The same thing like mast cell fiber plus mast cell fibers, we make a mast cell fascicle. And we know that tissue, many tissue combine together to form an organ. The same thing like skeletal muscle tissue, skeletal muscle tissue, we form skeletal muscle. And that skeletal muscle become an organ. So which means the pectoralis major muscle we saw, which is made of many skeletal muscle tissue, and therefore skeletal muscle, that is become an organ. And now organs plus organ is equal to system, which means our muscle and the bone, so one organ plus another organ, which we become musculoskeletal system. The same thing like the heart and the blood vessel will give us a cardiovascular system. There are 3 types of muscle: skeletal, cardiac, and smooth muscle. Skeletal muscle we will use for locomotor system. Cardiac muscle we use for cardiovascular system, and smooth muscle we use for involuntary movement organs like gastrointestinal tract. I want you to remember a few characteristics of skeletal muscle. They are contractility, excitability, elasticity, and extendability. This became important when we make a debridement of the contaminated muscle, because we want to remove as much as we can to make the muscle clean, but at the same time, we want to leave as much muscle as possible. Then if we want to check whether muscle is still viable or not, of course, if we make a cut, there will be bleeding. That indicates muscle has still blood supply. If we stimulate that muscle contractibility, electro-excitability, and if we stretch it, it is extensible and it is elastic. So that means that muscle is still viable. We want to preserve as much as we can. So now look at the pectoralis major muscle in the dead body, cadaver. And we can see these are the brown. And if you pull it, you won't be able to pull it. There is no more elasticity or extendability. And there will be no more contractility. And these are the pectoralis major muscles here on the chest. And this is the deltoid muscle, which is on the shoulder, which gives us the shoulder contour. Now I'm going to show you the muscles in the back. And look at that. This is a triangular shaped muscle, which is what we call trapezius. And this is a big, broad muscle in the back. Therefore, we call it latissimus, large dorsi in the back, large muscle in the back, latissimus dorsi muscles. Look at the extent of these muscles. Like this two muscle is nearly covering almost all the entire back of our body. So now we use the electron microscope and we start seeing 2Z, and this structure is called sarcomere, S-A-R-C-O-M-E-R-E, sarcomere. So now we need to look at the sarcomere, which is in drawing in detail, which will be like that. And these are the fibers which is running in between 2Z line, and that is what we see here. These lines are actually called myofilaments, which are actin and myosin. If you look at carefully, the thick, broad band, which is colored in red, that is myosin. And thin, small little blue color line, that is the actin. Actin is thin and myosin is big. Actin is thin filament and myosin is thick filament. So now, if we connect multiple sarcomeres in a linear fashion, so one sarcomere here, another sarcomere here, another sarcomere here, multiple sarcomere in linear fashion, that is called myofibrils. Myofibrils, a group of myofibrils wrapped around with the sarcolemma. Sarcolemma is the cell membrane for the master cell. And now you wrap again with the endomysium. This structure is called mast cell fiber, which is equivalent to cell, because it has got the cytoplasm equivalent, which is called sarcolemma. And now multiple mast cell fibers wrapped by another connective tissue, which is called perimysium. This is what we call mast cell fascicles. Mast cell fascicle combined together and wrapped around with another connective tissue, which is called epimysium. This is the mast cell organ. So the pectoralis major muscle we saw, the bicep muscle we saw, it is the organ, it is the mast cell organ, which is covered with the epimysium. So let's look at the little train in the Alp, Swiss Alp. So now it's one individual coach for a train is something we will consider as sarcomere. Inside the sarcomere, there are a few seats. They will be equivalent to myofilaments. So multiple sarcomeres arranged in linear fashion, linear direction. That is what we call myofibrils. Myofibrils, a group of myofibrils together, which is wrapped around with the sarcolemma, which is equivalent to the cell membrane. And that is again covered with the endomysium. That is what we call a mast cell fiber. Mast cell fiber is equivalent to cell because it has now cell membrane, which is what we call sarcolemma in the mast cell. So many mast cell fibers, which is enclosed by perimysium, is mast cell fascicle, and many mast cell fascicle enclosed by epimysium is the mast cell organ, which is something we can see and we can feel and we can touch. So the way I try to remember is Sacramento, which is equivalent to sarcomere to start with, and rail, little rail train, myofiber rail, and then mast cell fiber, ER, and then fascist. So mast cell fascicle, fascist, and muscular, that is mast cell organ. So now we know that myofilaments inside the sarcomere, multiple sarcomeres arranged in linear fashion is called myofibrils. Myofibrils together, mast cell fiber, fibers together, fascicle, fascicle become mast cell. So this is the seniority of the organization, which we should remember as the anatomy student. So now look at that. This organization, so all these epimysium, perimysium, and endomysium, they gradually fuse together, they put more collagen fiber, and they go and attach to the bone. That is what we call a tendon. So a tendon is very different from a mast cell, because mast cell is the composite tissue, which is made of mast cell cells wrapped around by the endoperi and epimysium, which are connective tissue. But when it comes to the tendon, it is more like connective tissue. If we look mast cell under the microscope, and therefore we are going to see some connective tissue, which are epimysium, perimysium, might be endomysium, but the red little stretches are mast cell cells and mast cell fibers. So why do we need mast cells? What are the functions of the mast cell? Of course, they produce movement. They open and close the passageway. For example, like those external anus sphincters. And when the time is not right, they will squeeze it. And when the time is right, they will relax and let it go. So they open or close the passageway. And they maintain the posture and stabilize the joint. For example, like the muscles in the back, like the muscles we saw, trapezius and latissimus dorsi, they are contracting all day long as long as we are standing upright or sitting upright. They have to contract. They maintain our posture. And muscle, when it contracts, it uses ATP and it produces heat. Few little terminologies here. I said cytoplasmic membrane is equivalent to sarcolemma. Cytoplasm is equal to sarcoplasm. So these are the mast cells, sarcomere, try to translate into our normal cell. Cytoplasm, sarcoplasm, we call it in mast cell. Cytoplasmic membrane, we call it sarcolemma. Endoplasmic reticulum, in the mast cell, we call it sarcoplasmic reticulum. And at the same time, what I want you to remember is sarcomere is the basic functional unit of the mast cell organ. The neuron is the functional unit of the nervous tissue, and osteon is the functional unit of the bone. So these are the functional units which we need to remember, and we'll be talking about this again and again. And as we say, mast cell is a composite tissue which is made of mast cell tissue plus connective tissue. These endo, peri, epi, they are all connective tissue. And of course, there will be capillary meshwork in the endomysium. Let's talk about a muscle origin and insertion. A muscle will arise from somewhere, usually from a bone, and ends at some point, usually in a bone, other part of the body, or it will gradually change into fascia and might go and attach to another muscle. So which is the origin, which is the insertion? It's easy to say, the more proximal portion of the proximal attachment is origin and distal attachment is insertion. But in our anatomy, we will also say the attachment of the muscle on the less mobile bone is origin, attachment of the muscle on the more mobile bone is insertion. So now if we look at this diagram, this is the humerus, this muscle originating from the humerus and become a tendon and go and attach to the proximal ulna. And if we do the flexion of the elbow, and if we flex our forearm, the forearm flex and mobile, the humerus is in stationary position. And therefore, by definition, this is the insertion, this is the origin. So now look at this photograph. This is in the same person. We are looking at the bicep muscle. On the right side, this muscle is small, rolled up, and bulge out. There is no distal extension. On the left side, this bicep muscle is still quite broad and bulky and still going into the elbow. What does it mean is this muscle and tendon fiber units, which is going to the elbow on the right side, has been ruptured. So this is the typical photograph of a distal bicep tendon rupture. On this is on the right side. So if we explore that area, we see the bicep tendon, which is ruptured, floating around, which we will have to stitch it back. So now we have taken a muscle fiber. So one muscle fiber here, another muscle fiber is running here. Now we have a good microscope, electron microscope, and we start seeing the single muscle fiber, which are made of multiple myofibrils. And now looking at the multiple myofibrils, which is nothing more than a longitudinally arranged sarcomeres. Now, if we look on the sarcomeres, that is what we see: myofilament, thin actin, and thick myosin. And as you know, the thick myosin does not go and attach to the Z line. Thin myosin attached to the Z, originate from Z line, but does not come and attach to the M line. And myosin attached to the Z only via these little yellow structures, which are called titin filaments. So now if we go deeper into that diagram, we are doing a cross-section in the different areas of the sarcomere. If you look at this area of sarcomere, we don't have any myosin fiber, we only have actin and this titin protein. So now if you look at only the actin area in a cross section or transverse section, you can see small little blue dots here. These are the thin actin dots, which is kept in the cross section. So let's look at this area, which is only myosin. Now you can see a bigger red myosin fiber, which are kept in cross section. So now if you look at the M M line area. In the M line area, these myosin filaments are connected to each other by accessory protein, like a spider web here. And if we look in this area where we have the both actin and myosin fibers, now we have the spot of blue actin and big rounded myosin area here. So these are the cross section at the different site of the sarcomere. We are all talking about this mast cell fiber. So these are the mast cell fibers, 1, 2, 3, 4, running straight, nice and parallel. They look striated. And now if we look at the single mast cell fiber, and this is what we are going to see. And the striations, because they have darker A band and lighter I band. And now if we look more detail, then we will see myofibrils. And if we look at the myofibril, which is nothing more than the longitudinal arrangement of sarcomere. And if we look at the single sarcomere here, this is what we see. We can see the 2Z. So this is in between 2Z is one sarcomere. There will be another sarcomere here. There will be another sarcomere here, and on and on and on. And now we start seeing the I band and A band and M line. And what are they? So if you look at the I band area, in I band area, we only have actin, and therefore it is light color. And the way I try to remember is I is small little thin, very thin structure, and in thin structure we only have thin actin. So if you look at the H zone, in the H zone, it is more in the center. We only have myosin. So the way I try to remember is H is double the width of I, and therefore in the H region we have myosin, which is much bigger than actin here. In the M line, as we saw, like myosin fibers are attaching to each other with protein elements. And if you look at the area in A band included the both actin and myosin fibers here, And therefore I remember as all, A for all, meaning both actin and myosin. I is thin, slim actin. And H, white, big myosin. And now we can start seeing better the titin protein filaments. They look like a spiral structure, like a spring. They attach the myosin to the Z disc. So now we are all clear of these anatomy. So if you look at the actin and myosin, then this is thin filament, which we call actin, and this is the thick filament, what we call myosin. If you look at the myosin has a small little heads. These are called myosin heads. Look at that. When we talk about actin, thin filament, when we talk about thin filament, we have three structures. They are actin, which is colored with blue. They are troponin, and there will be tropomyosin. So now you can appreciate thick filament, which is made of myosin and myosin head. And you can see the thin filament, which is made of actin, which is the blue structure, and troponin. These are the yellow little areas there. And tropomyosin, which is color with the yellow strand here. That is tropomyosin. We'll talk about these structures a little bit more detail later in the lecture. So now this is the revision. A for all, both actin and myosin. Z is the end. M is in the middle. I is thin, and therefore actin or thin filament. H is white, about twice the size of thin, and therefore it is myosin or thick filament. And this is the cross section, and that's the way we try to remember. So now when we see the sarcomere here, and these sarcomeres are wrapped together by sarcoplasmic reticulum. Look at that. These are the blue structures, which is not vein. They are called sarcoplasmic reticulum. So they run along with the actin and myosin. And when it gets close to here, then they go into different direction, horizontal direction or transverse direction. That is what we call terminal systems. So this sarcoplasmic reticulum, which is equivalent to endoplasmic reticulum, which will restore loads of calcium, sarcoplasmic reticulum, and the terminal systems, they are all similar structures, they all contain calcium. So this is one terminal system from one side. And now we can see another terminal system from one side. In between, we have a T-tubule, which is nothing more than the invagination of this white sarcolemma, which is the cell membrane equivalent, going deep into the sarcomere. So now we know that sarcoplasmic reticulum is longitudinal, terminal system is going horizontal. They both restore calcium, loss of calcium. T10 protein, T10 filament, it runs from the Z disc to thick filament, which is myosin. It keeps the myosin in place. It is like a spring-like structure which folds and unfolds depending on whether muscle is stretched or relaxed. As we know, sarcoplasmic reticulum is equivalent to endoplasmic reticulum. It runs along longitudinally with the myofilaments, and the perpendicular structure is called terminal system. And in between, we have transverse tubules. There are two types of muscle contraction, which is described in our textbook, which is concentric contraction, which means the muscle gets shortened, like when we flex our elbow, the bicep muscle shorten, and eccentric contraction, when we stretch our elbow, the bicep muscle lengthen. And there is another type of muscle contraction, which I haven't written here, which is isometric contraction. Iso means same, the same. Metric means length. Isometric contraction, same length. So muscle is contracting, but its length of fiber doesn't change. So now let's look at the driving forces, driving structures of the flexion and extension. We got thick filament, which is made of myosin with beads, myosin head. And we have the thin filament, which is made of blue actin, yellow tropomyosin, and these little patches, what we call troponin. So we'll look a little bit more detail there. So now on this side, this is this muscle in the relaxation stage. So now you can see the little beads of actin. They have a sweet spot where the myosin heads wants to come and attach for the mast cell contraction. These sites are called myosin binding sites. They are present on the actin. And when the mast cell is relaxed, this tropomyosin here, which is going spiral around these actins, tropomyosins are blocking and covering the myosin binding site on the actin. And we have a structure called troponin, which sits very close to the tropomyosin. Once there is a stimulus, when there is a acetylcholine release, and there is calcium release from the sarcoplasmic reticulum, and that calcium will come and bind to this troponin. And now look at that. Calcium has bind to troponin. By doing so, there is deformation of the tropomyosin. Look at that. This curvy, wavy tropomyosin becomes straight. In other words, we call this conformational changes. By doing conformational changes, these little spots, myosin binding site on the actins, get exposed. Now this myosin head is happy. They will go and attach to the myosin binding site, and that way start the contraction of the mast cell. And when there is no more calcium, this tropomyosin will go back to its normal shape, curvy shape. This myosin binding site will be blocked, and now mast cell will relax. So now let's look at the sarcomere level. What is happening here? So in this diagram, if you look at these titin filaments, they are long. And this is in a state, mast cell is relaxed. And these two Z lines are far apart. Now, once there is a actin and myosin contraction beginning, now these two Z lines get close to each other. Now the thin filament, which is mainly made of actin, began crossing the medial M line. And if you look at that, this spring-like titin protein gets shortened as well. This is in a state of mast cell contraction, and this is in a state of mast cell relaxation. Mast cell contraction, mast cell relaxation, and this is mast cell contraction. So now we look at the mast cell and the nerve. So this is our spinal cord, and this is anterior horn, this is posterior horn. This color is darker, so we will call it gray matter. This color is whiter, so we call it white matter. And look at the anterior horn. The nerve motor neuron come out. The purple color, it go attached to the mast cell fiber. And look at another motor neuron from the front anterior horn. This red color coded will go to different mast cell fibers. So here, what we need to remember is a motor neuron and all the mast cell fibers it innervates. So for the red 1, 2, 3, together with this motor neuron, that is what we call motor unit. And motor unit is the motor neuron plus the mast cell fibers it's innervate. So now this is the nerve ending. These are the terminal boutonnieres. They will come and act on the mast cell, and at the junction, acetylcholine is released. This acetylcholine will start the stimulus and causes sarcolemma to produce calcium, and that calcium will go to the troponin. Troponin will make the conformation changes in the tropomyosin. Tropomyosin conformation changes will expose the myosin binding site on the actin, and then now myosin head will come and attach to that site. That is the mast cell contraction. And when we are talking about these, I want you to remember these two free of charge websites, Auto Bullets and med bullets. These are the sites where they are useful, like especially for those aiming for medical school or nursing school. These are very useful websites which are free of charge. And now it comes to classification. Of course, we like to classify everything. We like to classify everything. And if you look at that, how can we classify mast cell fibers? Depending on how they make energy, which is ATP, adenosine triphosphate, and how fast they break down the ATP, how quickly they contract, because mast cell contraction needs energy, and this energy comes from adenosine triphosphate, ATP. So yes, the way they make energy, if they use oxygen, these fibers will be called oxidative fibers. And if they use glycolysis and they do not use oxygen, these mast cell fibers will be called glycolytic fibers. And the same thing, like if mast cell contract fast, or if they break down the ATP very quick for mast cell contraction, they will be called fast fibers. If they are slow to contract, then they will be called slow fibers. So in the slow, if we combine these two together, slow fibers are only oxidative fibers. Slow fibers are only oxidative fibers. Slow fibers are only oxidative fibers. But fast contracting mast cell fibers could be either oxidative or glycolytic, or might be a little bit of combination. So now let's look at this. Let's start with the slow oxidative fibers, which is slow and they only use oxygen because they need lots of oxygen. So they need a lot of blood flow. So therefore they need a lot of capillaries so that they pick up a lot of blood. Blood will contain RBC, which will carries oxygen. And that oxygen need to be trapped by the mast cells. And therefore this we use myoglobin. Myoglobin is the pigment which binds oxygen. So these many capillaries, we have many RBCs, many RBC will carry many oxygen. That oxygen will be entrapped by the myoglobin, and therefore slow oxidative fiber has lots and lots of myoglobins. And these myoglobins, we entrap the oxygen and we give it to the mitochondria, which is the energy house of a cell. And this mitochondria will produce energy. And these slow fibers are thin and small fibers. They are slow in contraction, but they tend to last for a long time. So the mast cells in our back, which is like trapezius, latissimus dorsi, or the mast cells for the spine, they are fatigue resistant. As long as we are standing up, they will be contracting. They will not get tired that easily. These are slow oxidative fibers. So let's go to the fast oxidative fibers. Oxidative fibers, of course, although they contract fast, they are still using oxygen. So the same thing like slow oxidative fibers, loss of myoglobins, loss of capillaries, loss of oxygen consumption, and they use the mitochondria to produce. But these fast oxidative fibers are, to some extent, they are fatigue resistant, but not as much as the slow oxidative. Their mast cell fiber size are intermediate in size, but they are faster contraction comparing to the slow oxidative fibers. So the example is the mast cell fibers in our thigh or in our legs. They can contract quick and we can run, but to good extent, they are fatigue resistant, but they are not like our back muscle, like spine muscle, because the back and spine muscle will be contracting all day long. we cannot be running all day long. Our muscles will get fatigued because they are fast oxidative. And now we come to the fast glycolytic muscle. They are using glycolysis for energy. Therefore, they do not need oxygen, and therefore they become anaerobic. They have very small number of mitochondria. They don't usually need loss of capillaries. They don't usually need myoglobin. They use glycosome and glycogen and breakdown. Their muscle fibers are pale looking, like our muscles in the breast, like chicken breast. It is pale color, not a brown meat. And they are fast contracting. Like look at the muscles in our fingers. They contract very, very fast. Close your hands, open your hands. You can do that very quick, but you won't be able to do this for 1 hour because they are easy to fatigue. So now look at this. And this is the cross section of 3 types of muscle: loss of myoglobin, slow oxidative, little bit in between, fast oxidative. They still have myoglobin, but they are not as much as slow oxidative. And fast glycolytic fibers, these are the white, pale looking fibers, because they will use glycolysis, they will not use oxygen, they do not have myoglobin, therefore their color is pale. Some clinical practice: rhabdomyolysis. Rhabdo means skeletal muscle, striated muscle, myo means muscle, lysis means breakdown. Rhabdomyolysis, the skeletal muscle breakdown. So for those patients, for example, like when they get drunk, lying on the floor for the whole night, whole day, next day, and all because of the pressure on their muscle, these muscles disintegrate and all the myoglobins come out from the muscle, which they go into the circulation and they block in the kidney, which can lead to renal failure. That is a process called rhabdomyolysis. Or the other example is burn injury to the muscle, or the muscle will become die and necrosis, and this will lead to rhabdomyolysis. And exercise and training can change our muscle, can make our muscles bigger. And with the training, we can change our thigh muscle into fast contracting muscle, like the sprinters, who run very fast on their legs. They are trained. Their muscles in the leg are trained to become like very fast contracting muscle. And there is a condition called muscular dystrophy. Dystrophy means dys means abnormal. Trophy means change in size. Dystrophy means abnormal change in size. And there is a condition called fibromyalgia. Fibro means fibrous, mya means muscle, algia means pain. All the muscle and fibrous tissue are painful. So it is a condition where very difficult to treat. They got pain here and there, which is a condition called fibromyalgia. So let's go and look at the muscular dystrophy here. So this is a form of muscular dystrophy. The name is Duchenne muscular dystrophy. It is very heartbreaking scene. Like they are pretty normal until the age about 2, might be 3, 4, 5. They look normal. So at the age of 1, the little baby is starting to walk around and you feel good. And starting from at the age of 2, their mobility gradually go down and gradually get worse. Look at this child. He is trying to stand up, but what he has to do is he has to be 4-leg fast. And then one of his hand need to put in front of the knee to strengthen the knee. And then both hands on both knee, he is trying to stand up and gradually lifting up his body, and now he is upright. That is what we call Gawa Sai. So that these patients with Duchenne muscular dystrophy, it only happens in male and it is X-linked recessive. And the problem is absence of dystrophin protein in the body. So it is very heartbreaking to see these patients, and they tend to die at the age of 20, 30 because of the respiratory failure, because their rib cage cannot move anymore for breathing. Few little things of note. Skeletal muscle will never undergo mitosis. So let's say if we cut a muscle, that muscle will not heal with muscle. Not like bone, that muscle will heal with scar tissue. The skeletal muscle will not regenerate. And androgen, testosterone, will make the muscle big. Therefore, male has bigger muscle comparing to female. And there is a condition which we call sarcopenia. Sarcopenia is usually age-related. It is a Greek word which we call sarco, mean flesh, mean muscle, flesh. Penia mean poor. So poor muscle. The muscle cells gradually fade away with the aging, or if somebody is immobile and just lying in bed, there is a condition called sarcopenia. And now look at this last diagram. Now his muscles are gradually getting smaller, which is a process of sarcopenia, and bicep muscles are getting smaller, and that is what we call sarcopenia. I'm going to end this lecture here, and I hope it is not too much for you guys. Thank you.